Today's lecture, we're continuing our discussion of vector-borne diseases with the genera Coxiella, Borrelia, and Bartonella. Like the Rickettsiales, Coxiella are obligate intracellular parasites and organisms which we aren't able to grow outside of cell culture. This is also an organism that we aren't able to visualize effectively using the gram stain. Coxiella has a worldwide distribution. It's found on all continents except for New Zealand. Borrelia are large spirochetes which have a small linear chromosome. This is the agent of Lyme disease, a condition which is increasingly recognized and increasingly surrounded with some controversy. Borrelia DNA has been found in museum specimens from ticks and mice from the late 1800s in Europe and the United States, so it has been found on both continents for over 100 years. Bartonella are small gram-negative coccobacilli, and if we consider the biocontainment levels of these organisms, both Borrelia and Bartonella species are level 2, while Coxiella burnetii is biocontainment level 3. Like I said, Coxiella doesn't grow outside of cell culture, and so cultivating it in vitro requires things like embryonated eggs in a high containment level facility. Coxiella is an obligate parasite, so it's host associated and it doesn't sort of live freely in the environment. It infects a wide variety of species, and it's shed in the milk, urine, feces, amniotic fluids at parturition. It is able to survive in the environment for 150 days, but it's not growing and replicating. It requires the host cells to do that. Borrelia are also obligate parasites, which are host-associated. We find it in both its reservoir hosts and also arthropod vectors. And it's interesting to know that Borrelia species are highly adapted to their specific arthropod vector. So the distribution of these pathogens is directly tied to where the vectors are located. Transmission is via tick bites, most likely the nymph stage. Bartonella are facultative intracellular parasites. We find them on erythrocytes and endothelial cells. These organisms also have principal animal reservoirs and then are oftentimes spread through vectors. So for instance, Bartonella henselae is one that we associate with cats. Up to 53% of cats have actually been shown to be seropositive, and it's spread by Tenosyphilides felis, the cat flea. Generally speaking, Bartonella cause a mild disease in their reservoir host, and we see pathology in more severe infections when an incidental host becomes infected. In this table here, you can see the vectors and suspected vectors for a wide variety of Bartonella species. This table was taken from this paper over here, Bartonella infections in humans, dogs, and cats. And you can see the wide variety of confirmed and suspected vectors for many of our different clinically relevant Bartonella species. Taxonomically, Coxiella only contains one species, Coxiella burnetii. There are 42 species of Borrelia and 39 species of Bartonella. When it comes to Borrelia taxonomy, we often group organisms by their association with Lyme disease. So Borrelia burgdorferi sensu stricto is the genetically defined Borrelia burgdorferi. This is based on its DNA sequence. It is that one particular species. Borrelia burgdorferi sensu lato includes a variety of other species which are all associated with Lyme disease. So genetically, they're different, but they cause the same clinical presentation. And so they're grouped together for that reason. With respect to virulence, relatively little is known about Coxiella burnetii. It's able to have phase variation with its LPS, so antigenic variation that helps to mask it from the immune system and avoid that antibody response. Borrelia burgdorferi produces outer surface proteins, or OSP proteins, which are important for attachment in the tick host and allow the organism to persist in the gut between meals. It produces surface lipoproteins, which stimulate inflammation and also persistence in ticks, porin-like proteins for adhesion, and then, of course, flagella for motility. Bartonella species produce type 4 secretion systems for effector secretion. and BEPs, Bartonella-associated proteins, which are secreted into host cells and really affect the physiology of those host cells in favor of the bacteria. So they do things like induce phagocytosis and prevent apoptosis to allow the bacteria in and then keep those host cells alive. More broadly speaking, there is evidence that bacteria which are vectored by invertebrates are able to change the behavior of those invertebrates to facilitate their persistence in the environment and transmission between species. So this was a really interesting paper, Pathogens Manipulating Tick Behavior. You can see here a variety of pathogens: Anaplasma, Babesia, Bartonella, Borrelia, Rickettsia, and all of the different things that they do to the tick to change how it acts and to improve their survival and transmission. Coxiella burnetii causes a variety of presentations in many species. In our small ruminants, we tend to associate it with abortions. In cats, we see infections which are most of the time subclinical, but can result in abortion. And then in people, it causes a disease that we call Q fever. Borrelia burgdorferi, so this is sensu lato. This is not just the genome species. It's all of those which cause Lyme disease. In dogs, cats, and horses is most commonly subclinical. No clinical signs are far, far, far more common than clinical Lyme. In people, it causes Lyme disease. Borrelia lone stori causes a Lyme-like disease in people, which is associated with the Lone Star tick, Amblyomma americanum. Borrelia afzelii causes Lyme disease in people in Europe, and Borrelia mayonii is a newly identified agent of Lyme disease in the last 5 years or so. There's a variety of Bartonella species which are clinically relevant. Bartonella henselae in dogs is associated with endocarditis and hepatitis. In cats, we see a similar spectrum of disease, but it's less common. And then in people, it's associated with what's called cat scratch disease. Bartonella vinsonii in dogs has been associated with endocarditis, myocarditis, and lymphadenitis. Bartonella ashlatica, which is found in wild rabbits, doesn't cause disease in animals, but has been associated with endocarditis in people. And then Bartonella quintana causes trench fever in people, and Bacilliformis causes carrion disease. In this table from the same paper that we discussed earlier, you can see the different species of Bartonella, the diseases they're associated with, and their geographic distribution. So Bartonella bacilliformis, the cause of carrion disease, is found in South America, while Bartonella henselae, which causes cat scratch disease and a variety of other syndromes, is found in South America, Europe, the US, Africa, and Asia. I think this provides a really nice summary of the disease organism associations within this genus. We're going to start by talking about Coxiella burnetii. This is a generalist parasite of eukaryotic cells, and it's found in many, many species. So from arthropods, reptiles, birds, and even mammals. It's the cause of Q fever, which is short for query fever. And this name arose from an outbreak of disease that was seen in abattoir workers in 1935 in Brisbane, Australia. So there was this unknown agent, hence query. When present, Coxiella burnetii is found in high concentrations in the placenta and fetal tissues, and animals frequently eat these tissues after birth. So it's birthed out, the animals eat those tissues, it passes through the gastrointestinal tract, it's excreted in the feces, it dries out, and can be aerosolized as a dust, which can then be breathed in by other susceptible animals. So aerosol transmission is very, very important for Coxiella burnetii. It enters the lungs once breathed in, replicates in pulmonary macrophages, and then is able to disseminate throughout the body. This organism has an interesting life cycle. There's multiple stages of Coxiella burnetii. So the small cell variants are taken up by those pulmonary macrophages. They're eaten. They are put into these vacuoles intracellularly. And when those vacuoles acidify, so when the macrophage tries to kill it, it stimulates the development of large cell variants. 2 days after infection, we have large cell variants replicating and the formation of what's called the parasitophorous vacuole. By day 6, we start to see the reemergence of these small cell variants. And by day 12, we have a cell chock full of small cell variants. It bursts, releases them into that extracellular space where they can go on to cause infections of other cells or transmission to other animals. In these electron micrographs, you can see Coxiella burnetii. On the left here, we have a vero cell. So this is a cell culture that's been infected with Coxiella burnetii. And you can see it is just chock-a-block full of organisms. And then on the right, we have a transmission electron micrograph of a Coxiella burnetii pure culture. Most often, infections with Coxiella burnetii are mild or non-apparent in domestic animals. Clinical signs related to these infections are really associated with where it localizes. So the reproductive tract and mammary tract. And we see abortions and then shedding of the organism in milk. So sporadic abortions are seen in our small ruminants and cattle, as well as potentially cats. We see placentitis and fetal pathology, including hepatitis, myocarditis, and interstitial pneumonia. Control of Coxiella burnetii outbreaks really relies on segregation of parturient ruminants. So you keep animals that are going to be giving birth separate from other animals, and then carefully disposing of tissues. So we've heard this before with other organisms that cause abortion. You want to remove those abortuses, and you want to remove all of those fetal membranes from the environment so that they can't be consumed by other animals or expose other animals. We also have vaccines available. There are inactivated vaccines, which can be useful in reducing the burden of disease. In this image here, you can see placentitis associated with Coxiella burnetii infection. And what's notable here are the multifocal, really pale areas associated with infarction. In dogs and cats, Coxiella burnetii infections are most commonly subclinical. If dogs are affected, splenomegaly is seen most commonly. And in cats, abortion has occurred. Interestingly, this has also been associated with human outbreaks. So we see cats sort of getting wrapped up in the same transmission events that are affecting people. And in affected animals, we see fever, lethargy, and anorexia 2 days following experimental infection. So the time course of disease is relatively rapid onset. In people, we term these infections Q fever. The incubation period is up to 3 weeks, and 50% of people remain asymptomatic, so it doesn't make everyone sick. Most often, it's an acute febrile illness, so kind of a flu-like illness, and it's typically a mild disease, although serious complications can occur. So we can see pneumonia, granulomatous hepatitis, myocarditis, and abortion. Among hospitalized patients, so if you're sick enough to end up in the hospital, there is a 2%, an approximately 2% case fatality rate. Back in the mid-2000s, there was a large multi-year outbreak in the Netherlands. Started in 2005 with the diagnosis of Q fever in dairy goats on 2 farms. In 2007, human infections were recognized to be spiking, and in total, over 4,000 human cases were ultimately identified. Among these patients, there was a 20% hospitalization rate, so a large burden on the healthcare system. And as a result of the subsequent epidemiological investigation, it was found that the most important risk factor for human infection was living less than 5 kilometers away from an infected dairy goat farm. And actually living downwind was very, very important. So this aerosol transmission was shown to be quite critical. There was also a cluster of infections associated with psychiatric patients who had really quite tragically had contact with lambs as part of animal-assisted therapy. The outbreak was ultimately brought under control through vaccination, manure management. So this was about removing feces from the farms, pasteurizing it, and not spreading it on fields. So stopping it from drying out and becoming aerosolized, essentially. Surveillance of bulk milk tank samples, and massive culling of animals. In Canada, Q fever is not a reportable disease. So this is some data from the US Centers for Disease Control. And you can see the number of cases that have been identified and reported from 2000 through 2019. So several hundred a year, although there does seem to be this sort of trend of increasing infections. In this map here, you can see state by state the annual incidence of Q fever. So incidence is the number of infections per unit population. And you can see it's highest in Idaho, the Dakotas, and Iowa, some of the most agriculturally intensive regions of the country. Q fever in people is acquired as an occupational disease, so it's unsurprisingly farmers, abattoir workers, veterinarians, and also laboratory workers. We have foodborne infections also recognized, so unpasteurized dairy products, particularly goat cheese, and there have rarely been associations with cats. We treat Q fever in people with doxycycline, and for high-risk workers, there is a vaccine available.